My name is Sarah Walters. I've been working for the Fish and Wildlife Research Institute for about 8 years. I'm working as a marine finfish biologist, so I study spawning in particular of spotted sea trout, snook, and redfish. So this is about as small as Seatrout will get in a spawning aggregation, but this is a male, and we know that because he already has his drumming muscles, and I can feel him drumming in my hand. This evening we are going out to target spawning adult spotted sea trout. We're going to capture them in a big seine net, so we're essentially going to scoop up everything that's there, the trout as well as little bait fish, maybe redfish, black drum, whatever happens to be in the area, but we're only pulling out the adult trout. And what we're doing is we're going to measure each fish, figure out if it's a male or female, which is really simple since they're in spawning condition. Just press on their bellies, and if it's a female you'll get eggs, and if it's a male you'll get milk. And then we take a fin clip back to the lab where our genetics team will be able to sequence the DNA to figure out the individual fish. And we're doing this simultaneously as another group at the FWRI, the Fisheries Independent Monitoring. They're capturing juvenile fish and collecting fin clips as well. And we want to see if we can track if those juveniles are coming from this particular spawning aggregation. And this particular aggregation is very unique for Tampa Bay. Typically, you'll find spawning aggregations all throughout Tampa Bay where you find them normally when you're fishing. But in this particular area, the fish seem to come every night throughout the whole spawning season, which usually starts in April and runs through mid-September. You don't usually think of them like you do kind of the typical residential spawning aggregations, like you think of the groupers, like in the Bahamas, where they all leave an area to go to a separate area for spawning. But we do know that these fish are not here during the daytime, and they're not here outside the spawning season for the most part. So we do see an active movement to this area, and we know that because we've captured them with nets. We fish when they're there and when they're not, and then we've also done a lot of acoustic telemetry where we've captured the fish, done a little surgery to implant a tag, and then left the receivers at this spot, and we see both females and males moving to and from the aggregation throughout the season. And unlike this special spot, we'd go back to some of these same aggregations and they would not be there every evening. This is one of the only spots that we feel like they're there repetitively. So we want to see, does this large group in this area disproportionately contribute genetically to the overall Tampa Bay population? But one other kind of byproduct of this study is that we're able to tell from this site, because we're sampling it twice a month on the new and the full moons, because we see an increase in fish during these times. We'll know if we're recapturing the same fish. From the genetics, we can tell if every time we're here, different individuals are coming, or if we're actually recapturing some of the same ones. And from that, you can kind of get a population estimate for this spot. At this particular spawning location, we saw an enormous impact from the red tide in 2005, as well as lots of other trout spawning areas and the trout population in general. And fishermen mirrored this as well in their catch. Everyone was having a really hard time finding fish. But since then, we've seen each year it getting higher and higher, and we're getting bigger catches and bigger fish as well. And that's in part thanks to trout. They mature at a really small size and age. So a little trout you'll get anywhere between 10 and 14 inches. He or she is probably able to spawn, and will do so in that season. So we've been really fortunate to see the trout come back nice and strong since that hit. If you'd like to know more about this research or other projects we have going on at the lab, go to research.myfwc.com.